Hello everyone. What we're going to talk about in this lecture is the third type of biomolecules, which are proteins. Biomolecules which are extremely important in living organisms because they do almost anything. You'll see later on throughout the course how many things they do. So in terms of organization of the lectures, first we'll talk about the function of proteins. You see how many functions they cover in living organisms. And then we'll talk about their structure. So we'll first talk about the building blocks, the monomers that make up proteins, which are amino acids. We'll talk about their structure, the common structure, and the differences, and we'll see how they're classified among themselves. And then we'll talk about proteins. So the structure of the proteins, we'll talk about all the four levels of structure: primary, secondary, tertiary, and quaternary structures. Then we'll talk about how some proteins, they need additional factors called cofactors in order to function properly. How some regions of proteins have specific functions and are referred to as domains. And then we'll talk about how proteins are folded or can be unfolded. How the structure of proteins can be done or undone, also through the help of proteins called chaperones. So what are proteins? Well, proteins, as you probably know, are polymers of amino acids, which are joined together through peptide bonds. This is the most diverse group of biological molecules, both structurally and in terms of functions. Structurally, they're made naturally by 20 different types of amino acids. So you can imagine the combination can create so many different types of proteins. And therefore, you can have them to do so many different functions. Now, in terms of function, here you have a list of what they do. As you know, 99.999% of the enzymatic reactions are mediated by protein enzymes. The other remaining part is referring to some RNA molecules that can work as enzymes on themselves. Then there are important structural functions. You see, if we talk about cytoskeletons and so on. They are important to move material across the membrane. They're important for mobility, forming like structure like flagella, for example, or to form, the fibers in your muscles or muscle cells. They have important regulatory function. They work as hormones, some of them. Others can actually receive the message from outside the cell, transfer inside the cells, working as receptors. Some other make antibodies and so on. So they have so many functions inside the cells, thanks to their incredible variability in terms of amino acid compositions. So all proteins are made by amino acid. And a few words about the amino acid, and then we'll get into the classification. First of all amino acids are identified by either the full name or the first letter, usually, if it's still available, if there are two of them that do share the first letter, or the first three letters of the name. So for example, glycine can be identified as a G or GLY as well. Methionine as an M and or MET. Now, structurally speaking, they all share a common structure, and they're different just in one group, which is referred to as the R group. So structurally speaking, you can imagine how they are all based, as you can see on the bottom picture here, how they are built, all basically starting with a molecule that could have been like methane, so a carbon with 4 hydrogen. And then you have that one hydrogen here would have been replaced by this group, the amino group. You remember it's a basic polar group here. And then the other hydrogen here would have been replaced by this carboxyl group. Now, the carbon, the central carbon here is referred to as the alpha carbon. And this is to distinguish it from any other carbons present here in the side chain. So there will be alpha, beta, if there are more, and so on. And so these amino groups, in order to differentiate them as well from other amino group or carboxyl group that are present in the side chain here, they're referred to as the alpha amino group. So all amino acids, they do have the central alpha carbon and one alpha amino group and one alpha carboxyl group present there. And they're all different in what is attached to the fourth binding site here of carbon. So this is what makes amino acid different. Now, they are classified into different groups based on what it is in their R group, the property of the R group. However, before I get in there, I want to stress one thing, that despite how they're defined here, they're defined as non-polar or non-polar. So all amino acids, in reality, they're polar. I get it all the time in students from later semesters telling me, oh, some amino acids are non-polar, thinking that if you put them in a polar solvent, they will not dissolve. No, they would all dissolve, okay? But some of them would dissolve better than others. Those that are polar would dissolve better than those that are classified as non-polar. But in reality, they're all polar because they all contain an alpha amino group and an alpha carboxy group, with both groups being polar. So bringing polarity to the overall amino acids, okay? So the classification instead refers only to what it is in the R side chain, the R group here, the variable one. So based on that, they can be grouped, first of all, into two groups, non-polar amino acids and polar amino acids. Then within the polar amino acid group, you can distinguish those that can acquire a charge, which are referred to as polar charged amino acids, and those that cannot acquire a charge, which are referred to as polar uncharged amino acids. And then finally, among those that can acquire the charge, and this charge is due to a proton binding or being released there, they can either lose a proton and become negatively charged, and in this case also, since they lose a proton, they are referred to as acidic. The other ones that can bind a proton, so they're referred to as basic amino acid, and they would turn positively charged once they bind the additional proton with its positive charge. So these are all the amino acids that are present in the non-polar amino acid group, so the very first group. As you can see, all of them are basically based on mostly hydrocarbons. Okay, so with the exception of cysteine here as a thiol group here, or sulfhydryl group as H. We talked about that, if you remember, when we talked about functional groups. Then you have tryptophan here, which would have a nitrogen in there too, or methionine here, which has a sulfur here. But all of them, they have elements, a carbon, hydrogen, that do bring very low polarity there. Okay, so they're non-polar, classified as non-polar amino acids. Now, the polar amino acids instead are divided as charged and uncharged. Here are the uncharged amino acids. So these amino acids now, they start containing some polar groups. This hydroxy group here, for example, there are groups here as well. This group here, this group, and so on. However, all of this group, they do not ionize. So basically what it means, they cannot release a proton, like this group here or this group would never release this hydrogen as a positive charged proton, and therefore would never acquire a charge. The same thing here, these groups, they cannot bind to protons, so they are not basic in this case. So they will never release a proton or bind a proton, they will never acquire a charge. They are defined as polar uncharged amino acids. Differently from these 5 amino acids here, which instead have groups within their R groups, which are carboxyl group, some case, or amino-derived groups here, so when it comes to the carboxyl group here, as you know, it's an acidic group, so it can release a proton, can dissociate releasing a proton. And when they do that, they would acquire a negative charge. So these are referred to as polar, negatively charged and acidic amino acids. Whereas these groups here, these 3 amino acids here, they have an R group which can bind a proton. Definition of what can bind a proton is a base. So these are referred to as amino acid, positively charged polar amino acid or basic amino acid. Okay. Now, These amino acids, moving to the protein structure now, they join together, and this is done at the ribosomes. You see during translation, we'll get into that in the second half of the semester. These are joined together, forming bonds through condensation reaction, so dehydration, sorry, reaction, which give origin to peptide bonds. Okay. The specific name here of this bond is peptide bond, in case I ask you, which is a direct bond in two different amino acids between what used to be the alpha carboxy group of the first amino acid and the alpha amino group of the second amino acid. Now, notice one thing. Some people like to depict the amino group here bound to the proteins as if it was a direct covalent bond. But this will mean, when you see a straight line, will mean a covalent bond. In reality, this is an electrostatic interaction. Okay, so this will be the most correct version to depict that. In any case, these two groups, the carboxy group and the amino group, can react together, forming a peptide bond and one water molecule. So is a dehydration reaction right there. When they do that, they form what is called a polypeptide. So a polypeptide is a series of amino acids joined together, like the one you see right here on the bottom. Now, all peptides, in all polypeptides, which are different from proteins, when you talk about polypeptides, you're talking about basically this amino acid attached to each other, this string amino acid. When you talk about proteins, you're talking about the overall structure with all the folds and so on, which has arisen from these polypeptides. So the three-dimensional structure there has arisen from the polypeptide. So when you're talking about these amino acid, these polypeptides here, you can always identify at the beginning what is called an N-terminal, which is where there is the free amino group, which is not involved in any peptide bond. And on the other end, that's where there is the free carboxyl group, which is referred to as the C-terminal of the polypeptide. Now, this is always shown in this orientation from the N-terminal to the C-terminal because this is the way in which polypeptides are actually assembled in the ribosome. So the first amino acid would have the second amino acid attached to the alpha carboxy group, not to the amino group. So the polypeptide will always start with a free amino acid and therefore an N-terminal, and would end up with a free carboxy group, therefore a C-terminal. Now, once polypeptides are formed, in reality they do not stay as a linear string of amino acids, but they do start forming structure. When we talk about proteins, you can identify four possible level of structure, which are hierarchical levels. What do we mean by hierarchical? It means that each one of them builds on top of the other. So they are all interconnected. You have the primary sequence, which is basically a unique sequence of amino acid, which is formed in the ribosomes, which then would form, based on the type of amino acids that are present there, will form the secondary structure. Here you have an example of an alpha helix. There are different types. And then based on the R groups you have here on the alpha helix, this secondary structure would further bend, forming a tertiary structure, which then could be the final structure of some proteins, but some proteins that instead are made by more than one polypeptide, so more than one of these strings of amino acids, each one of them leading to their own tertiary structure, like the protein here, hemoglobin. Now, when you have these different peptides joined together, or this different tertiary structure being joined together, you refer to as a quaternary structure. So you can see how all of these structures ultimately are interrelated to each other, and how changing one thing and one level will lead to the change of all the other structures. That's what happens with point mutation that change the DNA, and therefore in the gene, in one gene, could change one amino acid and have a completely different effect on the tertiary structure and ultimate quaternary structure eventually of a protein. So all these structures are hierarchical. Let's start with the primary structure. So the primary structure corresponds basically to the sequence of the amino acid, which is dictated originally by the gene in the genome, corresponding to that protein. And these, so this is the primary structure, okay? In the primary structure, again, you can identify the N terminal, which is at the beginning of the polypeptide, the C terminal, and then what is called the backbone. The backbone is basically the repeated sequence of amino group, the central alpha carbon, and what used to be the alpha carboxy group, then what used to be the alpha amino group, and so on. This string here is referred to as the backbone, which does not include the R groups, which do stick out from the backbone, okay? So if I talk later on, I talk about hydrogen bonds within the backbone, it means there are hydrogen bonds formed among the elements in this area here, not part of the R groups. So this primary structure, in reality, as soon as it's formed, it's immediately folded into a higher level of structure, which is referred to as secondary structure. Within the same polypeptide, you have different type of secondary structures. They're different depending on the shapes that they give origin to. Again, I stress there are many, many types of secondary structures. Okay, we always talk about the alpha helix and the beta pleated sheet, but there are many other structures, okay, they can find in proteins. And a protein can have alpha helix and beta pleated sheet, so many different secondary structures within itself. Now, no matter what type of secondary structure you're talking about, they are all formed only by one type of bond, which are hydrogen bonds, formed within the backbone, within elements on the backbone. And more specifically, they're formed between the hydrogen in the amino groups, alpha amino groups, what they used to be alpha amino groups, and the oxygen of what used to be the alpha carboxyl group. See better in the next slide what we're talking about. So a very common secondary structure, again, they're not the only types. Alpha helix and beta pleated sheet are the most common ones, but they are not the only type. So the common structure is the alpha helix. So now this is formed by twisting of the backbone, which will lead to the R groups being projected outward from this twisting. It's right-handed. So imagine take your right hand and the twist of your hand will go upwards in the direction of the thumb. It's different than if you look at the left end, it will be the other way around. So it's right-handed, always right-handed, this alpha helix, and it's made usually by a stretch of around 10 amino acid longs with a span of about 3.5 amino acid for each turn. Now, this is formed when you have a series of amino acid in the primary structure. So this will be the primary structure. Okay, imagine this being a primary structure, so a sequence, the polypeptide, where you have amino acid 1 here, 2, each R group obviously will be a different amino acid, 3, 4, 5, and so on. So now, depending on a pattern of bonding that you have, of hydrogen bonding happening there, you could get an alpha helix, or for example, a beta pleated sheet. So now, the bonds, as I said before, are within the backbone between the hydrogen bond to what used to be the alpha amino group and the oxygen bound to the carbon of what used to be the alpha carboxyl group. I say used to be because now these groups are involved in peptide bonds. So now, what pattern of bonds would lead to the alpha helix? So in this case, you have that each amino acid is binding to an amino acid which is 3-4 amino acid after. It's easier to see it than say it. So I'll show you here on the bottom. Look at the bottom. So imagine that now you have amino acid 3. So that's what the hydrogen from amino acid 3 forming the bond with the oxygen here in amino acid 7. So there is a distance of 4 amino acids here. So now what you would have to lead to a alpha helix, you would have a pattern which is basically repeated over and over, where you have, in this case now, amino acid 4 forming the bond with the amino acid, which is still 4 amino acids far. Now amino acid 5 then would bind to amino acid 9 and obviously 6 to 10. So all of them are binding to the amino acid, forming hydrogen bond with the amino acid, which is 4 amino acid further down in the polypeptide. So when you have a pattern like this will cause this twist, okay, right-handed twist, and which is often represented as a cylinder in protein structures when you see a higher structure, the overall structure of a protein, which is referred to as the alpha helix. Now, when you have another type of pattern, you can get another type of secondary structure, which is referred to as the beta pleated sheet. Now, in this case, this can be formed with anything between 3 to 10 amino acids. Is referred to as beta pleated sheet because if you look at the plane, in reality, these are not flat sequences, right? So every amino acid will form a plane, and when they bond, there is a current there. So when you form beta pleated sheet, now you have the planes are parallel to each other, side by side, at the same level, and even after they turn. So it looks like a piece of paper that's been folded several times. That's where the name comes from. This case too is formed, as I said before, all secondary structures are formed by hydrogen bonds within the backbone. But in this case, this is happening with a different pattern than the one that gives origin to the alpha helix. So look at the bottom now, and I hope you can see the difference. So now you have that basically this amino acid 3 is forming a hydrogen bond with amino acid 11. Amino acid 4 now would form it with the one before amino acid 11. And now 5 would do it to 9, and so on, 6 to 8, and so on. So as you can see, there is an opposite pattern there, right? And you can imagine how this basically would cause the folding of these polypeptide, right? Folding, imagine like if you're folding a piece of paper, which will lead to actually a pattern like this one, okay? Now, beta pleated sheet, depending on where they happen, or which sequences or where they are in the protein, can be defined as either parallel or anti-parallel. Your book doesn't talk about that, but it's important to understand when you talk about this. So when you talk about parallel beta sheets or beta strands, they're formed when the sequences are in the same direction. So imagine this being the polypeptide. So the direction is defined as N terminal to C terminal. Okay, so this will be the beginning of the polypeptide. This is the end. So now, if you have that this sequence here forms hydrogen bonds with this sequence here after, though, the sequence as folded, so they both will be N to C terminal. So here will be still N to C. So they are in the same direction, and these will form what is called a parallel beta strand or parallel beta pleated sheet. Okay, if they're opposite to each other, so in this case, this part of the sequence, which then forms hydrogen bond with this part here of the polypeptide, in this case, this is going from N to C, whereas this one is going from C to N. So they're opposite to each other. In this case, it will refer to as an anti-parallel beta pleated sheet or anti-parallel beta strands. Now, this has implication in the way also the bond is formed, and in general, the anti-parallel beta pleated sheets tend to form stronger structure overall. Okay. So these are the two main types of secondary structures: alpha helix, beta pleated sheet, parallel or anti-parallel. But there are many others. But they are all formed from hydrogen bonds within the backbone. Now, as a result of all of these type of bonds, though, you have to understand that once this bond happens, the R groups end up projected outside of these structures. And now, depending on the property of these R groups can actually start interacting with each other. So basically causing potentially a bending, for example, of these alpha helix. So for example, imagine these having a negative charge, this is a positive charge, they could actually start attracting each other. They could have a sulfide group, a SH, another one here, they actually form disulfide bridges. They would permanently bind to each other, causing the curve a curve happening here, right? A twist of this. So a twist of this will be defined as the tertiary structure, will lead, or the combination of all the twists of secondary structures, fold and twist and so on, will lead to the formation of what we call tertiary structure of proteins. Now, what type of bonds can happen between R groups? So only R groups now are responsible for the formation of tertiary structures. Which ones? So these R groups can form different type of bonds. I made an example. You can have ionic bonds. So if they have opposite charges in these R groups, for example, this is a basic group, this is an acidic group, negatively charged, positively charged. They can attract each other, causing the bending here. If there was an alpha helix, would have bent here, for example. Now you can form disulfide bridges. So two thiol groups or sulfhydryl group, SH, that would interact with each other, forming in this case a covalent bond. This is the only type of covalent bond which is involved in the formation of tertiary, and you see later on in quaternary structures. All the other ones are based on electrostatic, type of bonds. Other bonds you can have, hydrogen bonds. You can have hydrogen bonds, or you can have also a hydrophobic interaction, as it happens when we talk about lipids between the fatty acid tails. They form hydrophobic interaction. Same thing for the R groups. So these are the different types of bonds among only R groups. The backbone at this point is not important anymore. That cause the formation of these tertiary structures. And now, as a result of the final folding here of the secondary structure into tertiary structure, a polypeptide would acquire its final structure. A polypeptide, notice, okay? Not the protein. The protein can be made by more than one polypeptide. At that point, that will be the quaternary structure. But this could be the final structure of proteins made by a single polypeptide. And so this final structure, that's actually what will give the function to the proteins. So for example, this is an enzyme called lysozymes. It's often used to degrade bacterial cell walls. And so these, you have an alpha helix here, you would have some beta pleated sheets there, you can see that, and all folding together, they form this tertiary structure. You can see that disulfide bridges are important too in the formation of this tertiary structure as well. Now, this is what caused the formation, for example, what is called the active site of this enzyme, which is the site where it will bind the substrate and where it performs the reaction. And again, this can be formed only thanks to the formation of this tertiary structure. So primary, obviously first, secondary, and then tertiary structure. So the function of the proteins is dependent on the final tertiary structure. One thing I want to anticipate here, you see when we talk about enzymes and how, for example, pH, temperature can affect the activity of the enzyme. You can see how the temperature, the pH, can affect any of these bonds important for tertiary structures. So for example, pH could lead to this group to be ionized. So this group could lose the negative charge. At this point, this ionic bond would not exist anymore. All right. So temperature, just the temperature increase energy in the system. So even within the protein, so the energy could actually start breaking some of these bonds. And that's one of the principles why, one of the reasons why you can denature or get proteins to lose their structures by boiling them, right? You're increasing the temperature, you're giving energy, you break these bonds as well. So you can see how enzymes are sensitive to pH and temperature and how they can be also turned on and off, particularly by using pH. Like enzymes in your stomach will work at certain pH because they have this specific structure that they need at that pH. But then as soon as they move into the intestine from the stomach, the pH changes. That would affect their tertiary structure. They lose their function. Now, as I said before, tertiary structure can be the final structure of some proteins that are formed by a single polypeptides. Others are made by several polypeptides. And so the joining of these different polypeptides together is referred to as the quaternary structures. So only some proteins have quaternary structures. The bonds involved are the same that, sorry, I don't have it here on my slide, but they are the same type of bonds that are responsible for tertiary structure. So any of these bonds, ionic bonds, hydrogen bonds, hydrophobic interaction, disulfide bridges, in this case between R groups that are projected outside of the protein. So there will be a lot of R groups projected here, which can form the same type of bonds that we talked about before, but between different polypeptides. So when you have these type of bonds between different polypeptides, you talk about quaternary structures. Here you have an example of collagen. Collagen is formed by 3 polypeptides, which they're basically identical to each other, forming alpha helixes. And these alpha helixes, they twist, forming tertiary structure, the tertiary structure of each single polypeptide there. And then they join together through different type of interactions here. Again, interaction, similar bonds, similar to those involved in the tertiary structure. When they join together, they form the overall collagen fiber, so the quaternary structure of collagen, which is this one. Now, as you know, not all proteins do work on their own. Some proteins, most proteins I would say, many enzymes, they work on their own, but some others do need additional groups to be added to them. Otherwise, they cannot do their function. So these groups that are required for the function of different proteins to work, they're referred to as cofactor or prosthetic groups. Here you have an example on the right side. This will be hemoglobin. Hemoglobin is formed by 4 different polypeptides, 1, 2, 3, and 4, with 2 identical to each other, so 2 beta globin and 2 alpha globin, so 4 polypeptides together. And as you know, is that protein that is responsible for carrying oxygen and carbon dioxide throughout your body. Now, this is done, carrying of oxygen and carbon dioxide is not done through binding of oxygen to the polypeptide or the peptide here, but is done because hemoglobin, each subunit of hemoglobin contains a prosthetic group called the heme group, which has an iron here, which is actually the one that binds to the carbon dioxide. So oxygen and carbon dioxide will be bound here. Without this, that will not work. There will be no binding of oxygen and carbon dioxide. Now, so we talked about the structure, 4 levels of structure of proteins and so on. Now, within the overall structure of each protein, you can find regions which have specific functions, which are referred to as domains. They don't need necessarily to be connected in terms of polypeptide. For example, imagine this here. You could have the polypeptide starting here, forming some structure, then going to the other side, then going back here and going back and so on. But you could still identify on one side a region which has a certain function, so a certain domain, domain A, and here another region here which has a different function. So domains are defined based on functions. Now, they can be structurally and functionally different. There are many of them that are actually conserved. Some of them don't get reinvented from protein to protein, but they are reused from a protein to another one. One example of conserved domains are the transmembrane domains. Transmembrane domains are region, they're different. They usually are alpha helices. At least 25% of our alpha helices. They basically, so they have this right-handed coil, which has a specific sequence of amino acid, 10 amino acid, or 6, 7, sorry, amino acid, that all have R groups which are hydrophobic, non-polar, right? So by doing that, they allow this part of the protein, since it has non-polar R groups, to actually go inside the phospholipid bilayer in the cell membrane, which is non-polar, right? We'll talk about that even more when we talk about membranes. So that's a domain that oftentimes is very similar, almost identical, sometimes many times identical among different proteins that are found inside the cell membrane. Then there are other domains. Here you can see an example of a polymerase. This is DNA polymerase. One of the DNA polymerase. DNA polymerase can do 2 things. it can form DNA by reading, basically copying the DNA and copying one strand of the DNA into another one. We'll talk about that. And this is done by one domain. Whereas at the same time, if it senses that it has made a mistake adding a nucleotide, the same enzyme can actually go back, remove the nucleotides, and put the correct one. And this is done by a second domain that has what is called the exonuclease activity. The removal, at least, is the domain that does the has the exonuclease activity. So two regions in the protein that have different function referred to as two different domains of the polymerase. Finally, we talk about the folding of proteins, right? So, and how their function is dependent on that. Sometimes this structure, the structure of proteins can also be undone. This can be undone in biological system using pH, for example, or can be done in the lab, oftentimes using heat, like boiling proteins, or using chemical treatment. Beta-mercaptoethanol is a common chemical used to do this. And now what these do, they break all those bonds that are involved in the formation of the secondary, tertiary, and eventually quaternary structures. Now, depending on what you use, so if you use heat, you can basically break all the different type of bonds, hydrogen bonds, hydrophobic interaction, ionic interactions, except, and I stress, except for covalent bonds. So the peptide bonds will stay there, so the primary structure will not be affected by the heat treatment. And also the disulfide bridges, they are covalent bond. Heat cannot break them. So the disulfide bridges, so these bonds between thiol groups here, these sulfhydryl groups, could not be broken by heat. But they can be broken by chemicals. Beta-mercaptoethanol, as I mentioned before, is one of them. They could actually reduce, again, these groups for reforming the sulfhydryl groups from the disulfide bridges. So in this case, for example, if you take this protein, you do this, you treat it chemically, and with heat, you completely destroy the secondary, tertiary, and quaternary structure. In this case, only secondary and tertiary because it's a single polypeptide. However, you will not break off the primary structure, which is the sequence of these amino acids. And so now if you let them bring the protein, you let it cool down again, this protein actually will reform the bonds, will reform the disulfide bridges, and will reform most likely also the other bonds, the hydrophobic, hydrogen bond, and so on. And so we'll go through what is called the process of renaturation. So this was demonstrated for the first time in 1972 by Christian Affinstead, which actually demonstrated how overall the tertiary, up to the tertiary structure, as you say, and therefore the function of a protein ultimately is dependent on the sequence of amino acid. If you undo it and you just leave it there, at that point it can come back. If you remove the conditions of the naturations, then you know that the information is already present in the only structure that is left behind after the naturation, which will be the primary structure. So if you're in a situation just like this, just to be clear, right? That's what he showed, is that if you treat it with heat and chemicals, right, you bring it completely denaturated. So now there is only a primary structure left. But then you remove the chemicals and the heat, and this protein can reform its original structure and reacquire the function. So you demonstrate that the information ultimately to form the tertiary structure and therefore to acquire the function, it's all in the primary structure right there. And that's why Christian Affinstead got the Nobel Prize in 1972. Finally, last thing we're going to discuss here is how folding is done. So a lot of folding is actually done by the protein itself. Okay, so many polypeptides, they would actually reassemble and form the secondary structure and tertiary structure on their own after the formation of the primary structure through translation. However, there are also proteins that help with that. They double check that these structures are formed properly so that the function of those proteins can be carried on. And these proteins are called chaperone proteins, okay, or chaperonins here. These, what they do, they actually bind to the polypeptide and they help forming the secondary and tertiary and eventually quaternary structure. They work sometimes also with higher level of structure, like in the nucleosome. You see where DNA is wrapped around these histone proteins forming a nucleosome. Chaperone proteins control that as well to make sure that the DNA is properly wrapped around there too. So in this protein, oftentimes they are expressed at high level when there are stressing conditions, like for example, at high temperature. High temperature, as we say before, would affect the structure of a protein. And so you want to have also proteins that can allow this protein to retain their structure, to be expressed at high level. So chaperonins are expressed at a high level when the temperature gets high, and therefore they are often referred to, many of them, as heat shock proteins. So finally, what did we discuss in this lecture? Are a few things. We talked about, first of all, the function of the proteins, and we defined them as polymer of amino acids. Then we talked about amino acid. We talked about their generic structure and how they're classified based only on their R groups into polar, non-polar, charged, uncharged, charged, positively charged, negatively charged, acidic or basic. And then we talked about the structure of the proteins. So we went through the 4 levels of structure: primary, secondary, tertiary, and the quaternary structures. And we talked about the bonds that are important for their formation, how the hydrogen bonds between the backbone are in the secondary structures, whereas bonds, different type of bonds between the R groups, are important for the tertiary and eventually quaternary structures. And then we talked about cofactors, how some proteins need some cofactors, through the example of hemoglobin, how it needs the heme group to actually bind oxygen. So they need additional molecules, non-amino acid-based molecules, to carry their own function. Then we talked about domains, regions in proteins that have specific functions that oftentimes are conserved as well. And finally, we talk about the denaturation of proteins, or how they can be denatured with heat, chemical treatment, how they can reform their structure, and how some proteins, chaperonins, chaperones proteins, they do help with the folding of the acquisition, if you want, of the secondary, tertiary, and eventually quaternary structures of proteins.